I want you to try something for me. Just for a second, look at your hand. Or, your arm. Just really look at it. To the naked eye, you're looking at a surface, right? Maybe it's a bit dry, a few freckles, but it's basically a solid wall of skin.
It looks like a wall. It definitely acts like one. But if we could zoom in, really zoom in with a high-powered microscope, you wouldn't be seeing a static surface at all. You'd be staring at a jungle.
A microscopic jungle. And that is exactly what we are deep diving into today. We are decoding the skin microbiome. And we are not just talking about, hygiene or washing your face. We're basing this whole discussion on a huge review article from Nature Review's Microbiology, a brand new one from 2025.
And this paper it's pretty significant. It really shifts the paradigm. For a long, long time, the medical community look at bugs on the skin as either transient tourists,
just passing through.
Exactly. Or invaders, the bad guys. This paper argues they are neither. They're a complex, co-evolved ecosystem, arguably an organ in itself.
Wow. And it's not just bacteria, is it? I think most people have heard about good bacteria from yogurt commercials. But this paper mentions fungi, viruses, and mites.
Like Malassezia. You have viruses, we call them bacteriophages, that actually hunt the bacteria. And yes, you have Demodex mites living inside your hair follicles.
Okay, wait. Mites on my face.
And before you run to scrub your face off, the whole point of this deep dive is to understand why they're there. We want to move from that germs are gross idea to understanding how this ecosystem can actually be engineered to cure disease.
Okay, let's map this out then. The paper starts by just describing the environment itself. And honestly, it doesn't sound like a jungle at first. It sounds more like Mars.
It's a harsh place to live. If you zoom in, the skin is what we call a saline, acidic, nutrient-poor desert. It's dry, it's salty, it's not an easy place for life.
Let's break that down. The soil of this desert. We have the epidermis.
Right. And the outermost layer of that is the stratum corneum. To really get the microbiome, you have to understand this structure. Think of it like a brick wall.
Okay.
The bricks are dead cells called corneocytes. They're tough, they're flat, and they're filled with keratin.
And the mortar holding it all together?
That would be a lipid matrix. So fats, ceramides, cholesterol, the whole structure is designed to keep water in and pathogens out. So for a tiny bacterium, it's like trying to live on a dry, salty cliff face.
Life finds a way, because the paper mentions these oily oases.
Exactly. And that is the crucial distinction. Dotted all over this desert are these little holes, basically your hair follicles and sebaceous glands. And those glands pump out sebum, oil.
So in the middle of this desert, you've got these little oil wells.
And that drives everything. Which brings us to the first really big concept in the paper, site specificity. Your skin is not one uniform ecosystem. It's a series of totally different continents.
That makes total sense. My forehead is definitely greasier than my elbow.
And the bacterial map reflects that perfectly. The paper breaks it down into 3 zones. First, the oily sites. That's your face, your chest, your back. Here, the dominant citizen is Cutibacterium acnes, or C. acnes.
The acne bacteria.
We call it that, yeah. But in a healthy person, it's just the primary resident. It's a lipophile. It loves fat. It eats the sebum from those oil wells.
Okay, and then you have the moist and dry sites.
Right. Think of the crook of your elbow or your forearm. Here, the oil supply dries up. C. acnes populations they crash. And instead, you get way more diversity. You see Staphylococcus species, Corynebacterium species. It's a mixed neighborhood because the resources are so different.
And then there's the foot. The paper really seemed to treat the foot as like a complete outlier.
The foot is weird. It's physically so distant from the major oil glands on your upper body. But biologically, it's unique because of fungus. While Malassezia fungus is pretty much all over your body, the foot has a much, much wider diversity of different fungal species. It's basically an island with its own distinct flora.
So that's the geography. But landscapes change over time. The paper lays out a timeline for the microbiome that tracks with our age. And it starts at 0.
It starts at birth. In the womb, you're sterile. The moment your water breaks, the seeding begins. And this is where the research gets really granular about the mode of delivery.
Right, the C-section versus vaginal delivery debate.
It's not so much a debate as a biological divergence. If you're born vaginally, your skin is immediately coated in lactobacillus from the birth canal. They're the pioneers.
And if you're a C-section baby,
you bypass that whole canal. So your first colonizers are typically Staphylococcus species from the mother's skin.
Wow. Does that difference last forever? Are C-section babies permanently distinct?
The data suggests the communities start to look more similar around 6 weeks. But the diversity does stay lower in C-section infants for a while. But the real shift, the seismic shift that happens at puberty.
The oil boom.
Exactly. Hormones kick in, and those sebaceous glands go into overdrive. Suddenly, the desert is flooded with oil. This totally changes the food supply. The bacteria that love oil C. acnes, their population just explodes. They take over the face and torso. The community stabilizes into this oil-loving state.
Until we get old.
Right. Aging is essentially desertification. Sebum production drops. Sweat production drops. The skin physically gets thinner.
So the food source is gone.
And when the dominant species, C. acnes, starts to starve, it leaves a power vacuum. The stability collapses. The paper highlights that aging skin starts to pick up atypical Staphylococcus and other bacteria that normally couldn't compete in a healthy, oily, young ecosystem.
It's like a forest turning into a savanna. The big canopy trees die off and all the scrub brush moves in.
That's a perfect visual for it. And that instability is partly why elderly skin is so prone to infection and slow to heal.
Okay, so quick recap. Before we get into the mechanics, we're covered in the salty, acidic desert. The landscape is split into oily zones ruled by Cutibacterium and dry zones ruled by Staph. And this whole world evolves from a sterile slate at birth to an oil-fueled boomtown in puberty, and finally to a resource-scarce ghost town as we age.
That's a perfect synthesis of the terrain.
Okay, so we know who is there, but the big question, and the one that usually makes people a little uncomfortable, is what are they doing? Because my first instinct is to scrub them off. But the paper says these bugs have jobs.
Essential jobs. The paper calls it symbiosis. They are paying rent. And they pay in two main currencies, chemical construction and immune training.
Let's talk construction first. We mentioned the mortar of the skin wall earlier, the ceramides.
Right. We used to think the human body just made those on its own. But it turns out we have help. A bacterium called Staphylococcus epidermidis. We can just call it Staph epi. It secretes a specific enzyme. It's called sphingomyelins.
Sphingomyelins. That is a mouthful.
It is, but think of it as a catalyst. This enzyme helps process nutrients into ceramides. So this bacteria is literally manufacturing the glue that holds your skin cells together. You kill off the Staph epi, your skin barrier weakens, you lose moisture, you get dry and cracked.
So the bacteria is like a supply chain manager for the skin's structure.
Precisely. And C. acnes has a job too. It eats that sebum and breaks it down into short-chain fatty acids, or SCFAs, like propionic acid.
And acid means pH.
Correct. This is what maintains the acid mantle of the skin. Human skin is naturally acidic, around pH 5. Most pathogens, the really bad bacteria like Staph aureus or Streptococcus, they hate acid. They can't grow in it. So by producing this acid, C. acnes is effectively sanitizing your skin for you all the time.
Okay, that makes sense. They maintain the facility. But the second job you mentioned, immune education, that sounds a bit abstract. How does a bacteria teach my immune system anything?
It's like a training simulation. The paper talks about compartmentalized immunity. This is a crucial concept. Your skin bacteria are constantly interacting with your immune cells, but they do it in a way that creates a local response, not a whole body fever.
Give me a specific example of this training.
Let's go back to Staph epi. It interacts with a specific type of immune cell called a Treg cell, a regulatory T cell. In simple terms, Staph epi teaches the Treg cell to stand down. It teaches the immune system tolerance. It's saying, hey, I'm a friend, do not launch an inflammatory attack. Without that signal, your immune system might just attack everything, leading to constant inflammation.
But the paper also mentioned a window of opportunity for wound healing. This part, it actually scared me a little.
You're talking about the MA T cells, mucosal-associated invariant T cells.
Right. The paper says these are critical for repairing tissue.
They are. MA T cells rush to wounds to coordinate the healing process. But here's the kicker. The research shows that these cells only develop properly if you're exposed to specific microbes early in life, like riboflavin-producing bacteria.
And if you aren't?
The paper suggests the defect is permanent. If you miss that microbial window in childhood, you may never develop a robust population of these MAI T cells.
So if we keep our kids in a hypersterile bubble, we might be permanently deleting a piece of their immune software?
That is the implication. It lines up perfectly with the hygiene hypothesis, that a lack of microbial exposure is leading to dysregulated immune systems. We need the bugs to install the software.
That's heavy, but it's not all cooperation and teamwork, is it? The paper has a whole section on chemical warfare.
Oh, it's an absolute Game of Thrones down there.
Yeah.
It is a constant battle for territory.
And they have weapons?
They have sniper rifles. They're called bacteriocins. These are antimicrobial peptides that bacteria produce to kill other bacteria. For example, C. acnes makes a molecule called cutamycin.
Cutamycin.
It's a targeted toxin. It specifically suppresses Staphylococcus bacteria. It's C. acnes saying, this hair follicle is mine, stay out.
Yeah.
Conversely, another bacteria, Staphylococcus lugdunensis, makes lugdunin, which kills the dangerous Staph aureus.
And then there's this quorum sensing idea. I've heard of this in, computing, but how do bacteria do it?
It's chemical communication. Bacteria are constantly releasing these small signaling molecules. When the population density is low, the signal is weak, it just dissipates. But as they multiply, the signal gets loud. And once it hits a certain threshold, a quorum, it triggers a genetic switch.
A switch to do what?
Usually to attack, to release toxins or form a biofilm. It's a coordinated assault. They wait until they have the numbers.
That is terrifying.
But the good guys have a countermove. It's called quorum interference. Some of our friendly bacteria can produce molecules that jam the signal. They essentially cut the enemy's phone lines. The bad bacteria never realize they have the numbers to attack, so they just stay dormant.
It's biological electronic warfare.
It is. And it is happening on your face right now.
So just to sum up this job section, we've got construction, where bugs like Staph epi literally make the mortar for our skin barrier. We have education, where they train our T cells to tolerate friends and heal wounds. And if we miss that class in childhood, we might be in trouble. And then we have warfare, where they're using chemical snipers and signal jamming to keep the bad guys in check.
You've got it. It's a functional organ system.
But clearly the system fails. We get acne, we get eczema. The paper calls this dysbiosis.
Dysbiosis. It's not just an infection in the old sense, it's an ecosystem collapse.
Okay, let's look at eczema, atopic dermatitis. Millions of people have it. What is actually happening mechanically?
The villain is usually Staphylococcus aureus. In a healthy microbiome, Staph epi and other things keep S. aureus in check. In eczema, that check fails. S. aureus overgrows. But the mechanism of the itch is what's truly fascinating.
It's not just irritation.
No. S. aureus releases a specific enzyme called the V8 protease. Think of it as a pair of molecular scissors. These scissors go and cut a specific receptor on your nerve endings called PAR1.
Wait. So the bacteria is directly interacting with my nervous system.
Yes. By cutting that receptor, it fires the ITCH signal straight to your brain. It's hacking your nerves. And then, when you scratch, you break the skin barrier, which releases more nutrients for the bacteria to eat. It engineers its own buffet.
That is diabolical. Okay, what about acne? We know it involves C. acnes, but everyone has C. acnes. Why do some people get pimples and others don't?
It's all about the strain diversity. The paper points out that acne patients often have a loss of phylogenetic diversity. They have specific, you could say angry, strains of C. acnes.
Angry meaning what?
Meaning they produce way too many lipases. These are enzymes that break down your sebum into free fatty acids. And these fatty acids are highly irritating and inflammatory. So acne isn't an infection in the traditional sense. It's more of a metabolic imbalance. The bacteria are processing the oil incorrectly, causing chemical burns from the inside out.
And rosacea. This is the one with the mites.
The Demodex mites, we all have them.
Okay.
But in rosacea, their density just explodes. It's an overpopulation event. And interestingly, it might not even be the mite itself, but a bacteria called Bacillus oleronius that lives inside the mite that actually triggers the inflammation.
A bug inside, a bug on your face.
Welcome to biology.
Let's touch on the most serious one here. Cancer. The paper actually made a connection between the microbiome and skin cancer.
Yes, specifically squamous cell carcinoma. And it's a double-edged sword. On one hand, S. aureus seems to promote tumor growth by modulating our immune defenses, specifically beta defensins. It effectively hides the tumor from the immune system.
But the good guys fight back.
They do. A specific strain of S. epidermidis produces a molecule called 6-HAP. And this molecule inhibits DNA synthesis in tumor cells. It stops them from multiplying.
So we have bacteria that are naturally producing chemotherapy on our own skin.
Exactly.
This brings us to the so what section. We know the landscape, the jobs, the diseases, but the paper outlines a future of therapeutics, and it sounds like we're finally moving away from just antibiotics.
We have to. Antibiotics are like carpet bombing. If you take an antibiotic for acne, you're killing the Staph epi that builds your skin barrier and the bacteria that fight tumors. It's just too blunt. The future is precision sniping.
Okay, let's talk strategies. Strategy 1, bacteriotherapy.
This is using good bacteria to kill the bad bacteria. The paper highlights a specific hero strain.
Eschei 9.
Eschei 9.
Sounds like a robot.
It's a strain of Staphylococcus hominis. And scientists found that this specific strain produces things called antibiotics that kill S. aureus.
The eczema villain.
The eczema villain, right. But it leaves the other good bacteria completely alone.
So you could just apply a cream with SJ9 in it, and it would hunt down the eczema trigger without nuking the whole ecosystem.
That's the goal. Clinical trials are already looking at this.
Okay, strategy 2. Engineering. This is the synthetic biology part. We're not just finding good bugs, we're building them.
This is the most sci-fi section of the paper, for sure. Researchers have engineered S. epidermidis to secrete a tumor antigen.
Explain how that works.
Basically, they gave the bacteria a little flag that matches the cancer. The bacteria colonize your skin, they wave this flag, and it trains your T cells to recognize it. So when the T cells see the actual melanoma tumor later, they're already primed to attack. It's a living cancer vaccine that resides on your skin.
That is incredible. And what was the mention of Netherton syndrome?
Right. A genetic disease where the skin sheds too much because of an overactive enzyme. They've engineered a bacteria to release a protein inhibitor called lectii. But here's the cool part. They designed it to be smart. It only releases the drug when it detects the problem.
So it's a bioresponsive drug delivery system.
Exactly. And my personal favorite, the mosquito repellent.
Yes, please explain this. I need this in my life.
Mosquitoes are attracted to lactic acid on our skin, and lactic acid is produced by certain bacteria. So scientists engineered skin bacteria to produce less lactic acid.
So you become invisible to the mosquito.
In theory, yes. No more D-ties, sprays, just a little microbiome adjustment.
And finally, phage therapy.
The return of the virus. We can use bacteriophages viruses that only infect bacteria to target C. acnes. It's the ultimate sniper. It ignores human cells, ignores other bacteria, and just pops the acne bacteria.
So if we're looking at the horizon, we're moving from killing germs to managing a zoo. We have SJ9 as a smart bomb for eczema. We have engineered bacteria acting as cancer vaccines and mosquito cloaking devices. And we have phages as viral snipers.
That is the future of dermatology.
But there is a catch, right? The paper ends on this note. Personalization.
The one-size-fits-all problem. My microbiome is not your microbiome. My strains of C. acnes are different from yours. My immune system was trained differently.
So a generic probiotic lotion you buy at the store might not work for everyone.
It probably won't. The future isn't a single bottle of good bacteria. It's sequencing your specific skin map, identifying your specific missing links, maybe you lack SJ9 or you have too much S. aureus, and then designing a cocktail specifically for you.
It totally changes the definition of a cosmetic product. Your morning moisturizer might eventually be a living prescription.
It could be your vaccine, your antibiotic, and your anti-aging regimen. All alive, all self-replicating.
So we've unpacked a lot. We've gone from the desert landscape to the bacterial chemical wars, all the way to synthetic biology. I have one final question for you and for everyone listening. Knowing what we know now, that we have these MA T cells that need training and staph that fights cancer, does this mean we're too clean? Are we over-sanitizing our own ecosystem?
It's the big question, isn't it? We spent the 20th century learning how to kill germs. I think the 21st century will be about learning which ones to keep. Because if you sterilize the desert, you might just be wiping out your own defense force.
Something to think about the next time you reach for the hand sanitizer. That's it for this deep dive. Take care of the skin you're in and the millions of residents living on it. We'll see you next time.